Good evening, everybody. Thank you all for attending the second Translating Science webinar hosted by the Wound Healing Society Education Committee. If you have any questions throughout this evening's presentation, please use the Q&A box and we'll address as many questions as possible at the end of the webinar. And as a reminder, this webinar is being recorded and you will be able to review afterwards when it is posted on YouTube. I am Kath Bogie, the chair of the Education Committee, and I will now pass you over to Rummana Aslam, who will be moderating this evening.
It's good evening, everybody, and thank you for joining us. I'm Dr. Rummana Aslam. I'll be moderating the webinar today. I'm at Yale School of Medicine. I'm the Chief of Physical Medicine and Rehabilitation, Wound Medicine Director, and Program Director of a Wound Fellowship here. And it is my pleasure to pass it on to Marjana Tomic-Canic, who is going to be giving us a delightful presentation today, which I personally am very much looking forward to. On to you, Marjana.
Thank you, Rummana and Kath and everybody else. Welcome. And I want to thank Wound Healing Society for providing this wonderful opportunity to share some knowledge about wound healing and especially aspects of translation. My name is Marjana Tomic-Canic, and I am a professor and vice chair of research at the Frost Department of Dermatology and Cutaneous Surgery at University of Miami. I wear many hats, but one of them is directing wound healing and regenerative medicine research program. I also want to, I think relevant to this audience is that I've been past president of the Wound Healing Society and an active member for, I think, since I started my research career. Today, we will talk about preclinical testing for effective translation, mostly looking at different aspects of models, design, and reporting. So my outline is going to focus on very briefly about wound healing models in general. I will then focus primarily on preclinical models for testing. We will review different kinds of models with some practical examples and relevance to the actual patients. Discuss elements of the experimental design, and finally, at the end, I will share with you some new developments in developing guidelines for reporting of preclinical wound model experiments. So to this audience, probably not news, but wound healing is a complex process, as you know, orchestrated by multiple cell types that needs to be executed in a very precise and timely fashion. So there are multiple different cell types that contribute from different skin and compartments. In addition to that, obviously, there is a major crosstalk with microorganisms that by the barrier breach are entering the wound and contributing to this communication. Overall, the wound healing process many times in our lives executes properly, completes the closure, restores the barrier, and remodels the scar. And of course, this goes on and on through our lifetime of organism. However, in the context of chronic wound environment, so this entire process does not execute properly. There is a large portion of cellular mismatched crosstalk where the signals are simply not properly shared between the cells, and the cells do not execute this program properly. So the question really, if we are talking about a translational aspect, is how can this be modeled? Again, multiple cell types, multiple time points, a lot of different interactions going on over a certain period of time. And looking at our skin, obviously, it's a lot different from the organism to organism. And so the main question that you start with is, what is the goal of the experiments? If it's scientific exploration to better understand the process, then there are so many models that I cannot possibly even summarize them, some of which are shown here, many models to choose from simple to complex organisms, including, for example, Drosophila, zebrafish, chicken, mouse, and so forth. This has yielded fundamental discoveries in understanding the process. And here I'm just showing published video from Dr. Paul Martin's lab of one of the early studies showing in Drosophila how macrophages actually crosstalk and function in the context of simple wounds. Again, there are very sophisticated tools that can be used, such as two-photon microscopes and so forth. And this is, again, a live animal that is put to sleep under the scope, wounded. And these are the live keratinocytes in this animal recorded from the different distances from the wound. And you could appreciate the migration of keratinocytes close to the wound edge and more proliferation as their distance. This was work by Valentina Greco and developed a model to study this. And of course, you can now, with much more sophisticated tools, map out different cells during this process while the animal is still alive and the wound healing process is occurring. So overall, this is just a small scope of what is out there. So there is tremendous amount of models to study wound healing. However, if your focus is to perform preclinical wound healing testing for potential clinical applications, then the number of the potential models it becomes narrow, right? In terms of really relevance to the actual human samples, because we are talking about clinical applications, which means this will be brought to patients. And so we can group it by simplicity of in vitro models, cell-based assays that are either 2 or 3-dimensional, ex vivo models, where we are using as a tissue-based assays, whether they're animal or human, and of course in vivo models, where we are using small animals, large animals, and of course human, which is not a model. And obviously the complexity from 2-dimensional in vitro cell culture all the way to the animal testing increases, so do costs. And of course, we can gain a lot more valuable information depending on which model we are using. But again, coming back to the complexity of a chronic wound microenvironment, which is a target for a lot of these therapies that are being tested, right? There is a lot of different dysregulated processes that we already have identified as the hallmarks of pathophysiology of a wound: decreased crosstalk, as I mentioned, lack of migration of keratinocytes, but there are hyperproliferation, inappropriate differentiation, infection, biofilm, so forth. There is a lot of protease, high protease environment, whether they're coming from eukaryotic or prokaryotics, fibrosis, hyperproliferative elements of the fibroblast and a dysregulated deposition of extracellular matrix, chronic inflammatory response, again, completely different than what we normally see in acute wounds. And so one needs to very quickly understand that we need to rely on a combination of animal and human models to really replicate some aspects of the human chronic wound phenotype. We have to accept that it's impossible to replicate everything we see in patients. And so when we review in vitro models, again, these are very, very simple assays that, many of us have used. When the cells are grown in the tissue culture dish and scratched, you can actually monitor their migration and proliferation. If you want only migration, then you eliminate the proliferation aspect. This can be quantified. It can be done manually, automated, a high throughput. Now there are a lot of different equipment that can actually test this. And you could obviously play around by adding and testing different compounds using this. And this is just one of the examples where we are targeting, these are primary human keratinocytes. They are treated with vehicle or a special glucocorticoid, dexamethasone, that is basically coupled with BSA so that it only targets one fraction, which is membranous fraction of glucocorticoid receptor, showing inhibition of cellular migration. So it's relatively simple, it's quick, it's reproducible, and it's very useful as the first elements of testing. Of course, the source of the cells can be different, right? I just mentioned and showed you primary human cells, but they can actually be coming from the patients. And this is one of the examples where we use primary cells isolated from a chronic wound before and after debridement. We generated primary cells, again, from 2 different wound locations and tested if they are migrating. And to no surprise, the cells that were collected from the wound sample pre-debridement from the chronic wound did not migrate properly, where the cells isolated from post-debridement actually migrated very similar to controls. And also their response to, for example, growth factor, one of which is we tested here as an example, VEGF. Also, the response on pre-debridement did not show high response, where the post-debridement actually did. So this is, again, the source of the cells that is being tested can come from the patients. And that is also a very useful screen for certain aspects of testing and applicability. Another component is when you want to test the crosstalk, and I'm not going to go or test migration. This is another form of migration assay where it's called Boydin chamber, where there is a membrane separating 2 compartments, and the cells are placed either in the top one, looking at how many of them will go through the membrane. Again, membrane can be controlled. The size of the pores can control. So there can be a mix of cells, but you can select them by size. Or you can place 2 different cell types in the top and the bottom and see how they cross-communicate and which one actually stimulates migration of the other. So again, 2-dimensional cell-based assays. Now we are we are going into more complex assays. This is 3-dimensional assay, again, considered an in vitro model, but now these primary cells that we isolate either from patients or from healthy human skin can now be replicated, and skin is a wonderful organ that can be reconstituted in 3-dimensional as a 3-dimensional organ. But the beauty of that, it can be wounded, and that wound, that organotypic culture of human skin would actually heal in the incubator. And so this is really something that is tremendously important, because it allows us to use different sources of human or animal cells, for that matter. But it also allows us to do genetic interventions, targeted genetic interventions in these cells. For example, we can generate organotypic culture of skin where we knock in or knock out a certain molecule, whether it's in keratinocytes, in fibroblasts, and so forth, and then see how that influences wound healing. So overall, this gives us a lot more flexibility in the sense, and because of this 3-dimensional organization, it is much more relevant to the actual wound healing process. Obviously, it is time consuming, and obviously this goes together with the costs that are definitely significantly higher than the just simple 2-dimensional model. I want to point out to you one organization that is actively looking to collaborate and disseminate the information. This is European Network for Skin Engineering and Modeling. They have a cost action. They have received funding to basically form this network, which is already up and running and had many meetings. They have 2 meetings a year where they exchange and share protocols of how to generate these models, isolate primary cells for different kind of skin testing, not only wound healing, but I just wanted to mention this might be some useful resource to you. Now, staying with the cell-based assay, as I mentioned, the source of these cells can differ. And so what we have done, for example, in collaboration with Dr. Jonathan Garlick's lab at Tufts, we have used the primary dermal fibroblasts from diabetic foot ulcers and healthy keratinocytes to generate organotypic culture. And then we wounded that skin and looked at how the wound healing progresses. Interestingly, it's very challenging to generate keratinocytes from chronic wounds. We can generate them, but it's very, very hard to propagate them. And there is a whole science behind it that I don't have time to go into. But it's important to emphasize that when we used diabetic foot ulcer fibroblasts with healthy keratinocytes, what happened is that these diabetic foot ulcer fibroblasts communicated with healthy cells above and instructed them to stop healing. So instead of healthy keratinocytes converting diabetic foot ulcer fibroblasts into healthy ones, the opposite occurred. And now this crosstalk between dermal fibroblasts with the from diabetic foot ulcers instructed keratinocytes above to actually behave like the ones in diabetic foot ulcer. And you could actually appreciate the inhibition of healing that is very visible, lacking epithelial closure in these organotypic cultures. So again, very useful for various types of testing. This is another example where we use the different genetic manipulations. We either overexpressed microRNA 193 in keratinocytes and generated organotypic culture, again, showing complete inhibition of migration and healing, and did the flip experiment where we generated keratinocytes with overexpressing microRNA with microRNA antagomer, which is suppressing microRNA, and 193 showing a promotion of healing, which is, again, the inverse experiment of the top one. Again, organotypic cultures can be used for different genetic manipulation of the source cells. And I will just briefly mention, I cannot go into this, the complexity of 3D skin models has been flourished by induced pluripotent stem cells technology, where you can, from these IPC cells, generate various types of skin cells coming from the same origin, and of course include neurons, include melanocytes and so forth, some even hair follicles and so forth. So again, complexity of these organotypic models is here to stay, and I think it's currently being developed in many labs. And I will not go into the details. This IPC technology can be used to reverse the phenotype of a non-healing. I will, again, not go into the details of this in the interest of time. Now we are shifting from these models into ex vivo model. That is basically collecting the tissue-based models, collecting tissue from either different genetic mouse models or mice, one can actually place a piece of skin from a mouse and monitor the explant. Migration from this explant into the dish and quantify or immunostain and so forth. This is an example of ADAM12 knockout that we have tested the migration. This was the assay developed by Pierre Colomb. And it's very useful when it is very difficult to wound the mice for various reasons, or mice do not live long and so forth, but you can have some excess skin where you can measure the at least migration and draw some conclusions about the wound healing process. But this is one that it's widely used assay, which is human ex vivo assay, where we basically collect the discarded human skin from plastic surgeries. We wound this skin. We form a standardized biopsy. This is a kind of the outer tissue, which is also standardized, usually 8 millimeter, 6 or 8 millimeter, and then there is a wound in the middle that is being made smaller, either 2 or 3 millimeter punch, and basically maintained on the air liquid interface, and the skin is, as wonderful organ that it is, can actually heal in the incubator over a certain period of time, and that can be followed by different measurements and different methods. And so in addition to that, this can be combined and made a little more complex by bringing infection or biofilm. And this is an example where we have shown that in ex vivo, a wound treated with the biofilm, this is again human skin ex vivo wound where we treated it with the Staph aureus biofilm, and we have shown that there is a lack of epithelialization and increase of specific microRNA that is triggered by presence of Staph aureus in infected wound. And so, again, this was correlated to the human samples of chronic wounds. But again, complexity of these models is depending on different and various experiments should be kept in mind. And this was done in collaboration with Rivka Isarov's lab, where you know, the assessment of this human ex vivo infected models were done very thoroughly. First of all, looking at a Staph aureus biofilm formation, quantification of the bacteria, as well as responses, first reepithelialization, and of course, expression of various factors that are participating in wound healing, inflammation, and so forth. So again, the ex vivo human model is very useful. Again, it has its own limitations without certain without circulation and flux of inflammatory cells and angiogenesis and then fat cells and so forth. So there are obvious limitations, but it's very useful for assessment of how human skin will react to certain challenges or treatments. And I'm just going to briefly mention, and I know Kath mentioned this in her talk, there are various models of building organ on a chip and body on the chip. And again, one can measure the responses of human cells in the context of various challenges, including wound healing in these either two-dimensional or three-dimensional organ on the chip or body on the chip. Best approach, of course, is combination of models, right? And this is, again, emphasizes the importance, if you are looking at a translational aspect, really, it's important to include human patient samples in this assessment. So this is just an example of acute human wound where we are looking at caveolin 1, and one can appreciate that there in migrating epithelium, there is no caveolin 1. In other words, it's down regulated in the human wound. And then when you look at the chronic wounds, this is non-healing venous leg ulcer, this is non-healing diabetic foot ulcer, different kind of immunostainings, but you can obviously see how strong expression of caveolin 1 is seen in contrast to the acute human wound. And I will not go into the entire mechanism because this was already published, but the important part is that what we have found is that their caveolin plays dual role. One is to facilitate inhibition of healing by enhancing the effects of glucocorticoid signaling, while at the same time sequesters growth factors from the membrane blocking their signaling. So there are two different mechanisms by which this can occur. And so to test this, obviously we started with in vitro model where we did scratches and blocked caveolin by methyl beta cyclodextrin, or knocked out caveolin from the cells, again showing the differences in migration. And then, of course, generated either tested it ex vivo treatments or generated organotypic cultures with caveolin knockout, showing stimulation of migration in both ways. So again, multiple different models initiated by findings from the patient samples and then looking specifically on that mechanistic aspect and showing it in multiple different models. Challenging, of course, in chronic wound therapeutic development is really that core processes, as I mentioned, of repair and regeneration are although conserved, the in vivo models provide valuable insights into these mechanisms. The FDA requirement for preclinical testing and validation in animal models. FDA requires this, although what they want is really safety aspects of that. Translation of findings from preclinical models to clinical use has been difficult. And again, this is because no single animal model captures the complexity of human chronic wound. And so we come to in vivo models, and this just it's impossible to summarize all of them, but they are either small animals, large animals, and obviously human. They can be non-diabetic and diabetic. They can have presence of infection or not, and there are various different combinations, whether we are talking about small animals, including mouse, rat, or rabbit. Murine models are most frequently used in this context, and then large animals, pig, dogs, cats, and horses, and so forth, but primarily for the practical purposes and in laboratory setting, usually it's the pig. And so, again, there are multiple ways of how these animals can be treated different in various aspects of wound healing process, and challenges of a chronic wound can be replicated in these models. And the one faces the first question when you want to do a wound experiment that translates to human is which type of wound to use. Again, there's superficial wounding such as tape stripping, where you basically just remove the top cornified layer of the skin, basically compromising just the barrier structure and nothing else. And we have shown, for example, that if you do that in a perforating to knockout animals, and infect them topically with the MRSA, what happened is there is a strong dissemination. Even with the just tape stripping, there is a strong dissemination of bacteria, and all animals actually die. So again, showing in this model that even the small challenge of the barrier can actually have lethal consequences when it's antimicrobial protein missing. Again, comparisons between excisional wounds. Another obviously type of the wound is excisional wound, and when you look at the human versus murine diabetic wound healing, clearly there is a difference between the type of the wound in that context. And in murine diabetic excisional wounds, mice heal by contraction, humans don't. And so we often use splinted model where we are splinting, you are either attaching silicone ring around the wound edge, excisional wound edge, and either suturing or gluing in different ways of doing this, but preventing this contraction so that you can actually test epithelialization. And so again, the diabetic phenotype can be genetic induced and so forth. So again, there are differences between obviously human and animal model. And this is one of the examples that I, again, even within the model, right? And now we are talking mouse model. Depending on which model you choose, you might actually get different effects. And so this was the primary testing of preclinical testing of the recombinant PDGF that showed that stimulates wound healing in diabetic mouse. These are excisional, non-splinted wounds. But interestingly, if you use the silicone splinted wound, then you actually do not see this effect of stimulation of healing. Again, this is approved therapy, and it helps a certain subset of patients. But I will say that again, the choice of the model really matters. And the major challenge that we face is that the diabetic mouse model. The diabetic mouse model is let me see. Is really more representative of an acute wound in diabetic patients when you compare it to diabetic foot ulcer. And so when faced with such phenotype, I would say you can actually add complexity by, this is a model that has been described by Manuela Martins-Green and her laboratory, where you can use older diabetic mouse and then manipulate by different ways leading to increased reactive oxygen species, combining it with infection, and then actually that will induce chronicity in the context of the mouse compared to just regular diabetic mouse that has slight delay in wound healing compared to the wild type. Again, the point is that eventually all of these mouse wounds will heal, obviously with a different level of complexity. That the time by which the healing is delayed and the amount is obviously different depending on the model. I should mention that there is a very useful model for those who study regeneration. It was developed by Mayumi Ito, and this, again, it's very large excisional wounds on the mouse back, and this shows that there is actual regeneration of the hair follicles in the middle of that wound that recapitulates a developmental process of really getting the new hair follicle. So again, very useful model to study regeneration. There are very specific aspects of this model that need to be followed. And here we come to the pig model, which is really probably the most representative of human wound healing in animal kingdom. It shares the type of healing. There is a correlation with pig and human studies more than rodents. And of course, the healing is by epithelialization rather than contraction, as I mentioned in the context of the murine models. Another advantage is that it's a large animal, and there are a lot of different wounds and different conditions that can be tested, different type of wounds and so forth. This model was actually developed in Miami by Dr. Bill Eaglestein, Pat Mertz, and Steve Davis and colleagues. And it's been very useful, and it's one of the requirements for the translation is one of the models needs to be done on pigs. And so again, the additional complexity can be added by inoculating these wounds with different biofilms. They can be bacterial quantification and so forth. Again, there is a lot of advantages that can be done. However, this is obviously very costly and not easy to maintain in the context of the facility and the infrastructure that needs to be put in place in order for these experiments to be executed. And lastly, let's not forget human volunteers, right? So this was an interesting study done by Maria Morasso's lab from NIAMS, where they have looked at skin human wounds of the same patient and their oral wounds where their gingiva was actually wounded, and they followed the healing and looked at the different mechanisms by which we heal in our mouth compared to our skin and so forth. But again, IRB approval is necessary, consent is required, and there are a lot of logistical challenges in this type of approach. However, it is very, very useful when it can happen. And so the other important part of in testing of wound healing is really how do we assess that process, right? From in vivo experiments, many use planimetry, where they photograph the wound over time and then calculate the size by naked eye or by some sophisticated digital mechanism, or histology, where we can actually assess multiple aspects of wound healing, not just closure, but also different composition of epithelialization, scarring, angiogenesis, and so forth. And of course, biomechanical aspects where we can test tensile strength and stiffness and so forth in terms of biomechanical properties of these healed wounds in animal models. And of course, these molecular assessments, again, so this was just kind of the basic elements, but now we are going into more complex. There are on a small scale molecular assessments that can be done by different immunostaining, different kind of testing in terms of, for example, Western protein levels of RNA, specific targeted RNA elements. Or it can be assessed on a larger scale by using different omics approaches like transcriptomics, whether we are talking RNA sequencing, cut and run, proteomic, lipidomic, fax analysis, and so forth, or high resolution. These are now much more sophisticated methods that are now brought by new developed technologies, such as single-cell RNA sequencing, where from the tissue, you can break it down to single-cell compartments and sort individual cell types, and then assess them on different way of omics. And the same thing for spatial transcriptomics, where you can actually look at a particular tissue and then select the certain regions and certain cell types that you want to assess in the context of, for example, wound. And I think these are the methods that really provide a lot more insights and knowledge about the process itself or anything that is being tested. And so this is one of the examples where we did bulk RNA sequencing of and compared skin acute wounds to diabetic foot ulcers. I'm not going to go into this because it's published, but it's important. We started with the human specimen, specimens from diabetic foot ulcers. We showed by genomic analysis that there is a downregulation of immune cell recruitment, and we have shown this by quantification in prospective set of patients looking at downregulation of numbers and decrease the numbers of neutrophils and macrophages as shown here. And we further analyzed and concluded that it's actually the FOXM1 molecule that is responsible for this blocking and decrease of recruitment. And then we actually went back to the mouse model. So starting with human, went back to the mouse model and basically provided inhibited FOXM1 pharmacologically in the mice and shown that we can actually recapitulate that aspect of the human diabetic foot ulcer phenotype in diabetic mouse. Again, note the combination of human and mouse when you know what specific mechanism you want to target. This combination is very, very useful. I would want to just share one more thing, which is a cautionary tale of epidermal growth factor therapy development for chronic wounds. In preclinical testing on multiple models, the recombinant human EGF have shown significant acceleration of wound healing. This was paper actually published back in 86. And However, when this was tested clinically, it didn't really show much of the effect. And fast forward many years later, our lab actually started to look at EGF receptor in chronic wounds, and it turns out that in patients, the receptor of EGF receptor is no longer at the membrane in the patients with chronic wounds. It's actually sequestered, as I mentioned previously, and these are different ways of testing this. So the point is that this downregulation and mislocalization of EGF receptor in patients, which was not replicated in mouse models and in animal models for testing recombinant human EGF as therapy, basically explains of why this therapy did not work. And now you can develop different ways of reconstituting this EGF receptor and sending it back to membrane, but that's not the topic of this talk. And so challenges in choosing this most appropriate model, choices, unfortunately, are often made based on costs, timeline, simplicity of manipulation, and access to facility, right? It's not about what you are testing. It's something that you know, mostly what would be the easiest and quickest way to find the result. And so, unfortunately, in our field, we don't have guidelines that will tell us how to select the models, which models exist, which type of methods to assess. And in review of literature, the level of reporting details of these experiments is really variable, and it's very, very hard to deconvolute how the actual wounding was done, how was it assessed, and so forth. And so it becomes very challenging to replicate the data that is published in wound healing literature. And so this part of lacking of guidelines in reporting was something that we took as a challenge. And as I mentioned, that's a major problem. And there are obviously many variables in a wound healing experiment, from the type of the animal, type of the wound, size of the wound, time point of selection, wound assessments, wound treatments, wound management, and many more variables obviously can influence the outcome. And if they are not being captured, then it's impossible to replicate the data. And so the wound care collaborative community has a GAP group, which I'm a member of. This is, again, a voluntary organization of all wound care stakeholders from academia, clinical, industry, and FDA. And so, you know, the organization basically said, well, is there any way you can standardize preclinical testing? And so, together with the Wound Healing Society approval and participation, we decided to develop this guiding document that will standardize both animal and human in vivo, ex vivo, and in vitro models for preclinical studies, again, that are relevant to human wound healing. We are not talking about the general, we are talking only about translation. And so this guidance document will provide some guiding principles and good practices for using of preclinical models in wound healing research. And I am happy to report that that document is completed now. It's in obviously in a peer review process. It's resubmitted in a revised form to the wound healing, wound repair and regeneration. But it's called, the acronym is RAPS, which stands for the Wound Reporting in Animal and Human Preclinical Studies, or RAPS guidelines. And this document contains a summary of major animal and human models that are used for preclinical testing, discussion of both pros and cons for each model, and importantly, a checklist for reporting the details of this experiment. And so we try to capture, again, not every experiment can be captured. So these are mostly standardized different variables that are typically used in a wound healing experiment. Capturing which animal wound model was used, wounding process, details of the wounding process, wound animal maintenance and monitoring, wound assessment and analysis. And so these guidelines, basically, there are multiple anticipated uses for this. It basically aims to guide wound healing researchers for accurate documentation and reporting of these experimental conditions to regulatory agencies, ensuring compliance with animal testing recommendations. The goal is also to standardize methods reporting in scientific journal to promote, obviously, consistency and reproducible findings. To provide guidance and assistance in designing wound healing preclinical studies. What do I mean by that? When you look at this guidelines and when you look at the list of different variables that are being captured, it prompts you to start thinking about your experiment. Which type of wound I'm going to use, which animal I'm going to use, how I'm going to collect, and how am I going to, what time point is more relevant to the experiment, to my goal of the experiments and so forth. And of course, the aim of this is also to help guide scientific reviews. Again, not everyone is expert in wound healing, but needs to assess the wound healing data. And this experimental design details can actually help with that assessment. And of course, the goal is not to exclude other different animal research experimental reporting, such as ARRIVE, for example, but it's designed to complement. And so I'm just giving you a flavor. This is, for example, the checklist of human ex vivo and organotypic 3D human wound model. And you, again, this is the idea of how the multiple different variables are being captured in this guideline document that will hopefully see the light of the day very soon. And so I hope you can help spread the word. Again, the goal is to have this checklist document as a PDF that can be filled and downloaded and used for all these purposes that I mentioned. And of course, it is anticipated that eventually scientific journals that are reporting on wound healing studies will adopt this in their author guidelines. And of course, the proof is in the pudding, as they say. So again, how much we utilize this document and this form will actually show how important it is to our field. And so to wrap up, I hope I kind of gave you an overview of how wound healing is actually a well-studied process, that there are multiple different models from which we can choose and with various level of complexity. We need to be aware and we need to recognize that there is always limited translation to human wound conditions, particularly when we are talking about pathology. Best approach is really combining multiple models to form a composite mechanistic insights regarding the topic and the study focus that you choose. And when using animal models or for preclinical testing, again, some level of relying on human tissue for guidance is really important because as I mentioned, most approaches will show efficacy in animal models, but that does not necessarily translate to human. And the more we test it preclinically, with this awareness in mind, I think the more effective we will be in translation. And with that, I want to thank you for your attention, and I'm happy to take questions.
Thank you, Marjana. That was a very, very helpful and a very good review. I don't see any questions in the Q&A, but I will open it to the attendees to ask questions. And while everybody's thinking of questions, I do have quite a few. So,
Sure.
first, I'm very glad, of course, to know about the guidelines. I think it will answer many questions for novice investigators as well as, experienced senior investigators. I know you said it's close to being published, hopefully. Timeline-wise, you think few months?
Peer review cannot be controlled.
Yeah, exactly. We know that.
But I'm sure that, I'm hoping by the end of the year or maybe early next year that it will be published. But I'm thinking probably, since this is now middle of October, probably towards the end of the year.
Very good to know. And I'm sure it will answer my question, where, I guess I was lucky enough to start with a very experienced, of course, world-renowned mentor, Dr. Thomas Hunt. So I didn't
Yes.
have to think much. But for anybody starting out in wound healing, what would be the best way to go? Should they start at a cellular level, animal model? What would be the best approach? Because as you know, most of the time, the limiting factor is going through the IRBs and the IACUC approvals for anything.
Yes.
So it can definitely discourage young investigators. So what would be I guess your guidelines will answer that, but for the audience today,
Yeah,
for us to disseminate this information.
Yeah, I think this is really important question. When you look at literature, it becomes really challenging. And I think the most important aspect is, what are you trying to achieve with your experiment, right? That is really what is going to drive the choice of the model, right?
Correct. Yeah.
If you are new in the field, right, I would strongly suggest reaching out to more experienced, look at the literature, look at who is in overall wound healing field closest to what you are trying to test, and reach out. Our colleagues are very helpful. Wound Healing Society has a lot of members, and beyond, I think in our field, we are very supportive of young investigators, and we are always happy to share the protocols or advice, right? And I think we are lucky that we live in where technological advances, where we can communicate in different ways with each other. And I would encourage a beginner in wound healing field, regardless of their previous research experiences, is to always team up with somebody who has done it before. Because again, the guidelines will help think about and create and design in kind of help narrow the list of experimental design aspects that are important to think about before you actually execute the experiment. But logistics of doing wound healing experiments, especially in the animal, I think are important, and again, getting all of that in line, there is always helpful. It's always helpful to reach out to colleagues for the guidance.
For a senior mentor, yeah. One of the things that at each webinar, it's amazing. I pick up something important, many things, but today I was very impressed and blown away by the fact when you showed that slide of the fibroblasts from a diabetic foot ulcer and you top it with healthy keratinocytes, that they become unhealthy. And I'm thinking, this is why skin grafts or cellular grafts fail. If you don't do a thorough debridement down to like living tissue, which we know biofilms exist in subcutaneous tissue also. And we actually did a few series where, even though we think, and I would like a comment on that, is we do debride in office a lot of time, but I don't think it's enough in many wounds. So I've been sending them to the OR for the few surgeons who love to debride wounds, which are few, but they do, and they go really, really deep. And then when you put, a biological cover, many chronic wounds do very well. So I think
Yes,
that, go ahead.
I think, yes. I think that there are aspects of all of this that are kind of good and bad news, right? The most important part is, yes, especially in the context of diabetic foot ulcers, the debridement is really very, very important, and it's a standard of care. However, again, the extent of it and how well it's done, it's always a question, right? But I think that the good news is that when the wound bed is properly prepared, I think that there is, again, by removal of these non-healing cells, I think that there are receptive cells that can actually respond positively from the stimuli that are being provided with whatever choice of therapy, advanced therapy you may decide to use. But but in a sense, what's important take-home message is that the phenotype, the non-healing phenotype, can be reversed, right? So I think that even, it's hard and it's not easy and we have to work on it. But in general, I think this is one of the really important take-home messages that we have done. We have done, again, different studies of showing that these cells from the non-healing wound edge and wound base can be reversed and completely converted into something that looks a lot similar than acute wound, right? Because you actually want recapitulation of an acute human wound in a chronic wound environment.
Absolutely. And thank you for that. That was very enlightening. And then I would like to ask, for human studies, as you said, it's always a challenge, creating a wound, and people sitting in the IRB who don't know wound healing will go, no, what are you doing? But,
yes.
But how do you approach human studies for samples? How would you think is the best
so
advice
again,
for us?
Yeah, so in the context of, again, I am in academic institutions, so this is limiting to that experience, I would say, is, the plastic voluntary surgeries of removal of excess of skin from breast reduction, abdominoplasties, and so forth, can be, this is considered a discarded specimen, right?
Correct.
As long as you do not need to capture anything clinically about the patient, including all the patient identifiers, this usually is a very simple IRB approval that goes through the expedite process because it's discarded and de-identified specimens, right?
Great point.
Which is part of this is the first part, but the other part is also important, and that is really finding a collaborator in surgery department or dermatology department that will actually be willing to provide these discarded specimens for the research purposes, right? So again, collaboration, I think, in general should be a take-home message in the wound healing field. It's too complex to be done alone.
Yeah, and I think you've done a lot of research, I believe, in taking the samples of debridement, like what you debride from the wound.
Yes. So from the chronic, but that again depends. It can be also considered as a discarded specimen, the debridement tissue, providing that you do not care about clinical outcomes.
Right.
If you do, right, if you do want to follow patients longitudinal, right, and collect multiple debridement samples, then you need a full IRB with the patient consent, right? So there are different levels of regulatory aspects that are really very important to secure before you can actually proceed.
Very good. Well, thank you so much. I see that our audience have stayed, but are there any questions, comments?
I think that was a great presentation. Thank you, Marjana. And as you emphasized, collaboration is key. So thank you to everybody who has attended this webinar series brought to you by the Wound Healing Society Society Education Committee and Wound Healing U. As a reminder, the recording will be available on the Wound Healing Society YouTube channel and the Wound Healing U website within the next few weeks, free and accessible to all. Please plan on joining us for the next webinar, Beyond the Lab to Standard of Care, Regulatory Aspects of Translation, which will feature speaker Carla Sigeb, and will be held on November 12th at 6 p.m. Eastern. Thank you, everybody, and have a good evening. Bye now.
Thank you. Take care. Bye-bye.
Bye-bye.